Now we are going to review for exam 4, which is covering chapters 11, 12, 13, 14, and 15. In chapter 11, we talked about DNA, the genetic material. Chapter 12 was how genes work. Chapter 13, we went over genomics and biotechnology. Chapter 14, we discussed evolution and natural selection. And chapter 15 is how we name living things. So in chapter 11, we went over DNA, the genetic material. And you can remember we discussed DNA structure. So DNA is comprised of subunits called nucleotides. Those are the individual pieces. And each individual nucleotide has 3 parts. They all have a phosphate, a sugar, and an organic base. Now these organic bases that you see in green are the parts that will determine whether or not they are going to bind to each other. So we have purine bases and pyrimidine bases. Our purine bases are large bases with a double ring structure, like you see here. This is 2 rings, so that's a purine. And our purines are adenine and guanine. Our pyrimidine bases are smaller bases with single rings, and these are going to be either cytosine or thymine. So do purines or pyrimidines have a smaller structure with a single ring? And do purines bind to purines, or do purine nucleotides bind to pyrimidine bases? Also, what kind of bonds connect the nucleotides to each other? So purines have a larger structure and pyrimidines have the smaller single ring structure. I told you guys in class to think about, do pyramids have a double base or just a single base? And pyramids have that single base at the bottom, and that might be a trick to help you remember which one are pyrimidines. The pyrimidines are the smaller single ring structures. So do purines bind to purines? No. You can see that our pyrimidines are matching up with our purines in both cases. You remember that trick that a student in class said, which was that cars go in garages and apples are in trees. So if you know that apples are in trees and adenine binds with thymine, then you know this purine is binding to a pyrimidine. Also, what kind of bonds are connecting those nucleotides? So we see these bonds here. What are those bonds? Those are hydrogen bonds. So we know that DNA is a double helix shape. And here you can see us zoomed in on those hydrogen bonds that are connecting our opposite bases. So here we have a pyrimidine connected to a purine and a pyrimidine connected to a purine. So these are our complementary strands of nucleotides. Are the strands twins? No. The 2 strands of DNA that form the double helix DNA molecule are complementary to each other. So they're not twins, they're not identical and matching, they're puzzle pieces that are completing each other. That means they are complementary. And I included this because we have 2 spellings of the word complementary. If you're just trying to flatter someone or give them a compliment, we would spell it with an I. But if we're spelling it with an E, it's used to complete or provide something lacking. So these are completing each other when they bind together. They are complementary strands. And the complementarity makes it possible for DNA to copy itself in preparation for cell division. But how does the DNA molecule copy itself? The 2 strands of DNA that form the double helix are, we've got a repeat slide here. But how does the DNA copy itself? We found out that it is actually through a method of semi-conservative replication, and this is just meaning replication that results in 2 copies of the DNA, but each copy has 1 original strand and 1 new strand that has just been synthesized. So you can see that happening here with the use of DNA polymerase. So what's the goal of DNA replication? DNA replication just means we want to replicate. We want to make a copy of our DNA. So new somatic cells can have all the genetic information that the parent somatic cells have. And where is this happening? It's happening in the nucleus of the cell. So we store all of our DNA in the nucleus. It makes sense that that's where we copy it as well. The process of DNA replication involves several enzymes, but the three big hitters are helicase, DNA polymerase, and DNA ligase. You remember that helicase is the one that unwinds the DNA to expose the templates, and this is what's creating a replication fork. DNA polymerase adds the correct complementary nucleotide to the growing daughter strand. So this is the one that's carrying in that new nucleotide and finding where it should attach. It's important to remember that we can only ever add nucleotides to the 3' end of an existing strand. So that means we are adding to the 3' end. So we're going in the 5' to 3' direction because we're going towards the 3' end. That's where the new pieces are showing up. DNA ligase comes in after the new nucleotides have been added, and it seals those fragments of DNA together. I added these images to help you remember. So helicase is like the one that is unwinding that double helix. It is opening it up. DNA polymerase is finding where do these puzzle piece nucleotides fit? What's the right nucleotide and where can I add it? And they put it in place. And our ligase enzymes are sealing everything together once the new strand has been formed so that it can actually stay together. At the replication fork, a primer must first be added to give a place for the DNA polymerase to start. So definitely remember that. In order for DNA polymerase to know where to start adding nucleotides, it has to find a primer to bind to. That is its start line. Using one template, DNA polymerase adds nucleotides to the free 3' end of both strands. And we call the two new synthesized strands the leading strand and the lagging strand. Our leading strand is the strand that receives new nucleotides in a smooth, uninterrupted, and continuous fashion. So you remember we had that picture where with the leading strand, we're just able to keep on adding because we're going in the direction that the DNA has been unzipped or opened up. Our lagging strand is the other strand, and it has a template that has a free 5' end. And because of that, directionality becomes a problem, because new nucleotides can only be added to the 3' end of an existing strand. So that means that the second daughter strand is assembled in segments. It has to be one chunk after another, kind of out of order and disorganized. After that, the segments will be joined together to form the lagging strand. So here you can see that action happening. We've got helicase unzipping the DNA. We've got a DNA polymerase looking for that primer, so it knows where to start. There's the primer. So now DNA polymerase knows that's the starting point. Once we've added those nucleotides to the 3' end, we will have our enzyme ligase come in and seal those nucleotides so that we can have our double helix structure with two complete strands, one that is old and one that is newly made. But replication is not always perfect. Mutation is a change in a cell's genetic message. So anytime we're changing what our genetic message is in our DNA, that's a mutation. And mutations can result from errors in replication or be caused by chemicals or radiation. They can also involve changes, additions, or deletions to nucleotides. So you could have just the wrong thing went in the wrong place, or something was added twice when one nucleotide is added twice when it should have been only there once, or you are missing a nucleotide completely and you have a deletion. These occur rarely, but they are also the ultimate source of variation and the raw material of evolution in all species. A change in the base pair sequence of a gene is called mutation. So remember that. Anytime we're talking about mutation, that's the change in the base pair sequence of a gene. Mutations can alter the genetic message and they can affect protein synthesis. Because most mutations occur randomly in a cell's DNA, most mutations are detrimental because they're not intended. The effect of a mutation depends on the identity of the cell where it occurs. So we know that mutations in somatic cells will not be passed to future generations. They will only be passed to the somatic cells that are derived from it. So that is like if we have a cut on our hand and we're making new cells, if there's a mutation there, that's not going to be passed on to our offspring because it has nothing to do with our sex cells. Our sex cells are our germline cells. And if we do have mutations in our germline cells, the mutations will be passed on to future generations. And that's also where our evolutionary change comes from, mutations in our germline cells. Some mutations alter the sequence of one or a few pairs of DNA nucleotides, and these are point mutations. So if we're just zooming in on a single point where a mutation has occurred, that's a point mutation. And we've got these three different kinds of point mutations. We have base substitution, which is going to just change the identity of a base. Basically, the wrong thing got put in. Or we have insertion, which adds a base that's not needed, or deletion, which removes a base that is needed. If any of those happen, we have a point mutation. But if the insertion or deletion throws the reading frame of the gene message out of register, then we have a frame shift mutation. And these can be extremely detrimental because the final protein intended by the message may be altered or not even made at all. In class, I talked about how you could imagine a frame shift mutation is kind of like whenever you miss one button and you're buttoning up your shirt and now suddenly all of your buttons are off. Everything is thrown out of alignment in a frame shift mutation. In chapter 12, we talked about how genes work. And the central dogma that we talked about is that all organisms use the same basic mechanism of reading and expressing genes. And that is that we have DNA, which we need to turn into RNA so that we can make our proteins. That is the central dogma, and all organisms are using the same base mechanism. In transcription, we are inside the nucleus, and we have DNA helicase unwinding just like we did in replication. So we're unwinding that double helix again. And now our RNA polymerase is going to add complementary nucleotides. So that makes sense. DNA polymerase was adding DNA nucleotides, but now we're trying to make an RNA copy of a DNA strand. So now we have RNA polymerase, and it is adding RNA nucleotides to the RNA copy of the DNA information. RNA polymerase is seeking a promoter sequence. So just like DNA polymerase was looking for a primer, RNA polymerase is looking for a promoter. RNA polymerase will start adding nucleotides once it has bound to the promoter sequence, which you can see right here. So it's just a specific sequence that the RNA polymerase will recognize as a starting point. And again, new nucleotides will always be added in the 5' to 3' direction, which means we are only adding nucleotides to the 3' end of a strand. To correctly read a gene, a cell must translate the information encoded in the DNA into the language of proteins. So that's where our RNA comes in handy. And translation follows rules called the genetic code. The mRNA is read in 3 nucleotide units called codons, and each codon corresponds to a particular amino acid. Genetic information passes from the genes in DNA to an RNA copy of the gene, which is a process called transcription. So that's what we just talked about happening in the nucleus. We are making an RNA copy of our DNA information. Then we go outside of the nucleus into the cytoplasm, and the RNA copy directs the sequential assembly of a chain of amino acids into a polypeptide chain, which will become a protein. This is our process called translation. So in transcription, we are transcribing our DNA into RNA. In translation, we are taking the information in that RNA and using it to create a polypeptide chain, which will become a protein. You can see that just basic summary here. We've got DNA going to RNA in transcription in the nucleus. And then we've got mRNA instructing which protein to make in translation, which is happening in the ribosomes. Also, when we're making our RNA, there are many different types of RNA, but the main ones that we're going to be using in transcription and translation are mRNA, rRNA, and tRNA. So any one of those could be the RNAs that are made to be used in translation. mRNA is our messenger RNA, and it provides the coding information that was transcribed. So this is the actual copy of our DNA information. Our rRNA is ribosomal RNA, and it is the component of the ribosome responsible for recognizing and binding to the incoming mRNA. So the mRNA will come in, and because ribosomal RNA is there, that means we recognize a spot to bind to. Then we have our transfer RNA. These are the ones that are carrying our amino acids to the ribosome so that they can be assembled into a polypeptide chain. You remember that all the transfer RNA line up together so that eventually there are so many chains of amino acids lined up that they're able to accessibly bind to each other and create a polypeptide. So this combination of these two processes is called gene expression because it's how we are expressing the genes. Gene expression basically refers to the use of information in DNA to direct the production of specific proteins. That is our gene expression. And here's that overview where we start out inside the nucleus, where transcription makes an RNA copy of the DNA information. That RNA will exit the nucleus, go into the cytoplasm, find a ribosome so that translation can occur, which is where we are taking that RNA information and using it to make the correct polypeptide chains. Here again, transcription's in the nucleus. We are copying the info from DNA to RNA. Translation, we are on a ribosome in the cytoplasm. We are using the mRNA instructions, which will bind to our ribosomal RNA on the ribosome, so that the mRNA codon can match up with all of the tRNA anticodons, which determines which amino acid is going to be added next to the growing polypeptide chain. So our codons are lining up with the anticodons. We have codons on our mRNA strand, and they are made to match perfectly with our anticodons on the tRNA. And that's how we end up with the right amino acid, because our tRNA will have a specific anticodon, which is what determines which amino acid is attached to it. And if that tRNA anticodon matches the complementary codon on mRNA, then that's how the complex is formed, so that we can eventually have this polypeptide chain. When we talk about the genetic code, it was determined from trial and error experiments to work out which codons matched with which amino acids. So just like we have the periodic table for our elements, we have the genetic code, which tells us which amino acids are matched to which codons. And the genetic code is universal. It's employed by all living things. It's not like we have one genetic code for humans and another for elephants. We all are using the same basic genetic code. Our genetic code is made up of 64 codons, and you'll remember that we had this table to break down which of those codons are coding for which amino acids. So for instance, if we have CUG, we start with the first letter, C, that puts us here. And then our next letter is U, so now we're going down this column. And our final letter is G, so that lands us here, which means it's in the block that codes for leucine, our leucine amino acid. We have 64 codons in total. A codon is a sequence of 3 nucleotides that can be found in either DNA or RNA. So I know that can be confusing, but it can be considered part of both molecules. It's just usually when we're discussing codons, it's going to be in the context of protein synthesis. So it usually refers to the codon sequence on the messenger RNA, which is transcribed from DNA. Codons and genes kind of sound the same whenever you look at the definition, but really, basically, this is the difference. Codons are nucleotide sequences of 3, and they are coding for a specific amino acid. So just one single amino acid is what that 3 nucleotide sequence of a codon is coding for. But genes are much longer nucleotide sequences, and they're coding for the entire specific protein. So every amino acid is included in the instructions of a gene. And that's just to help you break down what's the difference, because we hear codons, we hear genes, they're both coding for something, but the difference is codons are just coding for a piece of that genetic information in the protein, and genes are coding for the entire strand of a protein. Now let's match some complementary codons. So remember that trick, cars and garages, apples and trees. That means cytosine is going to bind with guanine and adenine is going to bind with thymine. But when we change from DNA to RNA, our thymine is replaced with uracil. So that's okay. That just means that anything that would have been T, we are replacing with U. So if we have our codons up here in teal, and then our anticodons are shown in purple, you can see that our A is pairing with what would have been T, but because this is RNA, now it's a U. A would have paired, or our U would have paired with, sorry, our U would have been T, would have been thymine, but because it's RNA, we don't have the thymine. So now we are having this sequence, and our guanine, like always, is pairing with our cytosine, and so on for these other examples as well. So to correctly read a gene, a cell must translate the information encoded in the DNA into the language of proteins. I think this is a repeat slide that got rearranged, so let's just use it to remember one more time that a codon is that 3-nucleotide sequence that is coding for a specific amino acid. And now let's talk about exons and introns. So you remember that eukaryotes, all eukaryotes have the coding portions of DNA nucleotide sequences that are interrupted by non-coding sections of DNA. The coding portions are known as exons, while the non-coding portions are known as introns. And I told you guys that my trick for remembering that was just that DNA exons, the information's actually exiting and getting expressed, whereas introns, the information is not going to code for a protein. But prokaryotic genes are just an uninterrupted stretch of coding DNA. So they don't have exons and introns. Every gene that they have is going to code for something, which makes sense because they're simpler. So in humans, exons may be spliced together in many different ways. And by using these different combinations of the exact same exons, different proteins can be created. This is termed alternative splicing. And that's what results in, we only have approximately 20,000 genes in the human genome that are encoding proteins, but we can still make like 100,000. Those are approximate numbers, but it's close. So that is how we were able to do it. We only have 20,000 genes, but because we can cut them and paste them together in all these different arrangements, we're ending up with upwards of 100,000 proteins that we're coding for. How prokaryotes control transcription. So every cell must be able to regulate when particular genes are used. And in prokaryotes, genes can be just turned off by the binding of a repressor, which is a protein that binds to a site on the DNA called the operator and blocks access to the promoter. So when we have a repressor come in and bind to the operator, which is this little section here, you can see it overlaps into the promoter section, which is what would need to be bound to for transcription to take place. So we've got something sitting in the way, basically. RNA polymerase can't do its job because this repressor is sitting on top of the operator, blocking the promoter. Genes can also be turned on by an activator, which is a protein that makes the promoter more accessible to RNA polymerase. Also, we talked about operons. And an operon is a segment of DNA in prokaryotes containing a cluster of genes that are all going to be transcribed as a unit. So yes, they're separate genes, but they will stick together. Anytime we're transcribing this LAC-Z, we're also going to be transcribing LAC-Y and LAC-A because these are part of an operon, which is a group of genes that are transcribed together. So the chromatin structure of eukaryotic chromosomes can be altered in several ways that will affect transcription. In chromatin, DNA is packaged around histone proteins to form nucleosomes. And modifying histones to result in greater condensation of the chromatin can actually make the promoters less accessible for gene transcription because now everything is wound so tight that you can't even get into transcribe. Also, methylation of the DNA can ensure that turned off genes will stay off because they no longer have the binding capacity. RNA molecules can also play a role in regulating gene expression. So we have these small interfering RNAs, which we call siRNAs, and they can block the transcription of genes whose sequence is complementary to the sRNA, called RNA interference. And this is also called gene silencing. I more often hear it referred to as gene silencing. So genes are silenced because the translation of mRNAs is blocked or mRNAs are destroyed. So here you can see our mRNA, which is ready to be transcribed, but because the siRNA is complementary and has bound to it, that gene has been silenced. There's nothing to be copied because it's not open and available for a copy to be made. In chapter 13, we went over genomics and biotechnology. Genomics is a field that compares the entire DNA content of different organisms. So the genome refers to all of the genetic information of an organism. That is all of its genes and the non-coding DNA as well. The genome includes exons and introns. So genomics is when we're talking about the field that's comparing the DNA of different organisms. And the genome is talking about all of the genetic information in one organism. DNA sequencing is a process that allows scientists to read each nucleotide in a strand of DNA. So that makes sense. DNA sequencing, you're looking for the sequence of nucleotides in your DNA. In DNA sequencing, heat is applied to denature the double strand because we've got to open that strand up. And the solution is then cooled to allow for the primers to bind to the single-stranded DNA. After that, DNA polymerase synthesizes complementary strands until a chemical tag is incorporated, and that will be the stop point for that copy. So the human genome, the publication of the sequence of the entire human genome occurred on June 26, 2000. And the human genome contains more than 3 billion base pairs. But it is estimated, like we said earlier, that there are 20,000 to 25,000 protein encoding genes. That's only 1% of our entire genome that is coding for all of the proteins that we need. There are also 4 major types of non-coding DNA, which account for 99% of the DNA in humans. Non-coding DNA within genes, those are introns. We also have structural DNA, which is just tightly coiled DNA that remains untranscribed. And then there are repeated or duplicated sequences. So there's simple sequence repeats and duplicated sequences that are scattered about chromosomes, and they're not coding for anything, they're just accidental repeats. We also have transposable elements, and these are bits of DNA that jump from one location on a chromosome to another. You remember in class we talked about how transposable elements made up 45% of our genome. So that's a huge part of bits of DNA that can just jump from one location on a chromosome to another. Genetic engineering is when we're moving genes from one organism to another. And the first stage in a genetic engineering experiment is that you have to chop up the source DNA and obtain a copy of the gene that you want to transfer. So restriction enzymes, which are also known as restriction endonucleases, are going to bind to specific short sequences on the DNA, and they will make a specific cut. That cut generates DNA fragments that are sticky because the incision made by the restriction enzyme is usually made to the side. It's not where those hydrogen bonds are connecting nucleotides. It's on the side of the strand. So those ends are now sticky and available to be bound again. When we move a gene or a part of a gene to a different location on the same chromosome or even onto a different chromosome, that is when we have made recombinant DNA. So it's recombined. It is recombinant DNA. So that is the process of recombination. Anytime you're taking one chunk from a gene and putting it somewhere else on that gene or on a different gene, you have made recombinant DNA through recombination. DNA from another source that is cut with the same restriction enzyme will have the same sticky ends. So these ends can very easily be joined together by the enzyme ligase. And restriction enzymes are the basic tools of genetic engineering. Now that we're on to genetic engineering, we have to add these restriction enzymes into our enzymes in action because they're the ones that are going to cut the DNA that we need to rearrange genes on. So forming our complementary DNA. To obtain DNA without introns, genetic engineers will first isolate the processed mRNA corresponding to a particular gene. So first, just get that piece of information that you need from the mRNA. And the enzyme reverse transcriptase can then make a complementary DNA version of that mRNA, which is called complementary DNA. So that makes sense, because if we are making an RNA copy from DNA, we're using, well, that's not the best way to say it, but I like to think of it like we're reversing transcription with reverse transcriptase, because instead of making RNA, we are making DNA from RNA, which is backwards. It's reverse. So that's how I remember reverse transcriptase. So our polymerase chain reaction, whenever researchers are going to make multiple copies of a gene without first inserting it into a bacterium, they employ this PCR. And we showed that really in-depth video in class. But this is where a double-stranded DNA fragment is heated to separate the strands, so we're denaturing again. And each of the strands is then copied by DNA polymerase. This cycle is repeated many times, over and over and over and over again, until we have so many samples of that DNA copy that was our target. We also discussed subunit vaccines, which are fascinating. So genetic engineering is also used to create subunit vaccines against viruses, like the herpes and hepatitis viruses. And in these, engineers are splicing genes from the coat of the target virus. So that would be either herpes or hepatitis, whatever we're trying to protect against. And we're taking those genes that are on the coat of the virus, on the outside, into a fragment of harmless cowpox virus, which is going to be our vector. That's what's going to carry this target gene into whoever is being inoculated. The recombinant virus will be injected into humans and cause the production of antibodies against the virus, which is essentially harmless to humans. So this method of using one virus as a vector to introduce fragments of the DNA of a disease-causing virus produces what we call piggyback vaccines, because we have one piece of that viral component that would cause us harm. It's just one piece, not the part that causes us harm, just the part that makes our immune system recognize it. And we're putting that into a harmless gene from a cowpox virus, and that's what makes it able to be inserted without causing harm. So genetic engineering. We also use this in agriculture, and engineering crops to be resistant to insect pest reduces the need to add insecticides to the environment. For example, we talked about Bacillus thuringiensis, which produces a protein that is toxic when eaten by crop pests. So these genes have been inserted into the chromosomes of tomatoes and cotton and lots of other plants. And because the plants can synthesize this Bt protein, now they're toxic to the pests that want to eat them, like the tomato hornworm. Aside from that, we also have herbicide resistance. So herbicide resistance is when we are genetically engineering some kind of resistance to an herbicide. So glyphosate is a powerful herbicide that kills most actively growing plants and is used to control weeds. Using a gene gun, engineers have been able to insert a gene into crop plants from a bacterium that is resistant to glyphosate. So they took that gene from the bacteria, they put it into these plants, and now they can spray their whole field, and their target crops are not going to be affected by glyphosate, but all of the weeds who don't have this protection gene from bacteria are going to die. The goal of genetically modified plants is to produce crops with desirable traits that directly benefit the consumer. For example, to combat iron and vitamin A micronutrient deficiencies among the world's rice eaters, genetic engineers created GM golden rice. And this is that transgenic rice we talked about that contains genes from a bean, a fungus, a wild rice, and a daffodil, all to increase its nutritional value. But the main concern about these methods of modifying the genes of our crops is whether or not these modifications are going to be harmful to other organisms in the food chain. When we talked about reproductive cloning, we discussed Campbell's colleague Wilmot, who attempted to transfer a nucleus from an adult cell into an enucleated egg, which is an egg with the nucleus removed. Wilmot used an adult sheep's mammary gland as the nuclear donor, and both the donor mammary cells and the enucleated eggs were first starved and then given a brief electrical shock, which allowed the contents to fuse together. The resulting embryo developed into a blastula and was implanted into a surrogate mother. And as a result, Dolly the cloned lamb was born on July 5th, 1996. So anytime we're talking about producing offspring by having changed the nucleus, that's reproductive cloning. In stem cell therapy, we have adult stem cells, which are pluripotent, and that is because they can differentiate into many different cell types, but not every cell type. Embryonic stem cells are the ones that can differentiate into any cell type in the body. That means they are totipotent, which is a great word that just means they have total potential to turn into any kind of cell, totipotent. When we discuss therapeutic cloning, that is where experiments are using stem cells to repair damaged tissues that have been carried out. First, we discussed experiments using stem cells to repair damaged tissues that have been carried out in mice without functioning immune systems, and it worked really well. But that's also because the mice didn't have functioning immune systems. So we have to overcome that. For stem cell therapy to work in humans, the problem of immune rejection needs to be addressed because we mostly do have functioning immune systems. So that's where therapeutic cloning comes into play. And therapeutic cloning, which is also called somatic cell nuclear transfer, addresses the issue of immune acceptance. In therapeutic cloning, the nucleus from a patient is inserted into the enucleated egg cell, and that egg cell develops in vitro. After that, the embryonic stem cells that have the same genetic information of the patient are removed and used to replace the patient's damaged or lost tissue. So that's a way to overcome this immune rejection, because now the new stem cells have the same genetic information. They're recognized as part of that patient already. So when we're using embryonic stem cells to restore damaged tissue, you remember that we will extract our embryonic stem cells and grow them in culture until we have our embryonic stem cells that are available to be reinserted into the patient. In nature, bacteria uses the CRISPR sequence to attack viruses by recognizing and cutting up their DNA. And using genetic engineering techniques, scientists can redirect the CRISPR sequence to recognize and destroy any 30-base sequence that they substitute for the spacer. So there's a great video for that on Canvas as well that you may or may not have seen if you were in class that day. Gene transfer therapy involves introducing healthy genes into cells that lack them. So gene transfer therapy is when we're taking healthy genes and putting them into the cells that don't have them. Early work with a cold virus vector called adenovirus was unsuccessful in humans because of immune attack. So they tried to use this virus, but it was too recognizable, and it didn't work. So then they selected a smaller vector, the adeno-associated virus, which is not going to be as easily recognized by an immune response. And it seems promising, but there are problems to overcome with gene therapy techniques. The vector used to carry the gene may elicit a strong immune response, just like what happened with our adenovirus. We could have too strong of an immune response, and now our target gene can't be introduced because the immune system killed the cell that was carrying it. And that can lead to rejection of the added gene. Or a virus vector may insert its DNA into the middle of a gene, which is not supposed to happen. Genes are supposed to be uninterrupted. They stick together. That's why they're genes. And so because it's been inserted in the wrong part of a gene, that can lead to cancer. So these are things we have to overcome before gene therapy can be as widely used as scientists would like. CRISPR is an exciting new discovery that permits scientists to edit genes. And the technology is based on an unusual repeating DNA sequence found naturally in bacteria. So during gene editing with CRISPR, Cas9 and CPF1 are going to make the double-stranded cuts in DNA at locations directed by our guide RNA. So these Cas9 and CPF1, those are our restriction enzymes that we're talking about with CRISPR, with the CRISPR complex, because they're the ones that are cutting. Our restriction enzymes are the scissors. Remember, they are cutting the strand in the right spot. And that is Cas9 and CPF1 with the CRISPR complex. In chapter 14, we talked about evolution and natural selection, and we got to discuss Charles Darwin's research. So Darwin voyaged from 1831 to 1836 on this long trip around the world. They spent most of their time around South America and a good portion of it in the Galapagos Islands, which you see right here. Darwin observed firsthand different plants and animals in various locales, and these observations played a very important role in the development of his thoughts about the nature of life on Earth and how everything developed. In 1859, Darwin published his writings that explained his theory for evolutionary change called natural selection. Darwin envisioned the frequency of favorable characteristics increasing in a population through a process called natural selection. So this is where favorable characteristics are specific to an environment, and they may not be favored, and they may be favored in one but not in another. So it's not necessarily happening to every species of every individual of a species population. Organisms whose characteristics are best suited to their particular environment will survive more often and leave more offspring. Which makes sense. If they're better suited to their environment, they're the ones that are going to be more successful at reproducing. Evolution is defined as the change in the DNA of a population over time. And Darwin's finches and the studies by the Grants, who also spent many, many years on the Galapagos Islands, provided evidence that differences in beak size were the result of natural selection and they were inherited characteristics. So we talked about Darwin's tenets of evolution, and the tenet is basically just a principle or a belief. So especially one of the main principles of a religion or a philosophy, it's usually used in that term, in that context. But according to Darwin's theory of natural selection, the key tenets are these. One, that variation exists within a population, which we can observe to be true. Two, that these variations are heritable, which means they can be inherited from parents of offspring. And then more offspring are going to be produced than can survive. His biggest example of this was the sea turtle hatchlings. But in all species, he's saying, more offspring can be produced than can survive. And finally, and most importantly, only those that are most suited to the conditions at the time survive to reproduce. So individuals with advantageous variations are more likely to survive and reproduce, leading to the gradual change in a population over time. Essentially, survival of the fittest, where beneficial traits are passed on to future generations through reproduction. Interestingly, we also talked about homologous and analogous structures. Our homologous structures are going to be similar structures in different species that are derived from the same body part present in an ancestor. For example, we've got this picture on the right where the same bones might be put to different uses in related species. So you can see we've got the same basic bones, but they're all serving different functions that are specific to these species' environments. And they were all derived from the same ancestor. So that's where that homo is. Homo is always meaning same. So homologous structures have the same ancestor and similar makeup. Analogous structures are structures that look similar, but they're actually in completely unrelated lineages. So that's just by happenstance of where they developed separately. They weren't actually related to a common ancestor. And this says, these are the result of parallel evolutionary adaptations to similar environments. So that makes sense. You ended up in a similar way, but you don't have the same ancestor if these are analogous structures. Genetic drift describes random changes in allele frequencies. So in small populations, the frequencies of particular alleles may be changed drastically by chance alone. And in extreme cases, individual alleles of a given gene may be all represented in just a few individuals, or even accidentally lost if those individuals fail to reproduce or die. So that can result in a genetic drift, like this. There used to be a gene in the population where some of these birds had a yellow phenotype expression, but due to genetic drift, we now are only producing this red phenotype in the remaining survivors in that species. So a species, we talked about how it's defined as a group of populations that are capable of interbreeding and producing viable offspring. So it's not just can you produce viable offspring, it's not just can you produce offspring, are they viable? Will they be able to one day reproduce as well? And this was the example I gave you guys of the liger, where yes, a tiger and a lion can reproduce together and create a liger, but that liger is not viable offspring. It is not able to then mate and create offspring because of this. So that is what makes them separate species. Speciation is the macroevolutionary process of forming a new species from a previously existing species. And this involves successive changes to the DNA. So our speciation is any time one species is formed from a different pre-existing species. We also talked about our barriers to reproduction, and this is called reproductive isolating mechanisms. And they cause reproductive isolation by preventing genetic change between species. So we discussed how there are prezygotic isolating mechanisms and postzygotic isolating mechanisms. Prezygotic just means before the zygote can even be formed, when the sperm and egg fuse together, before that even happens, these are the mechanisms that are preventing it from happening. Postzygotic isolating mechanisms are happening after the zygote has been formed. So still the genes that are being passed on are being isolated, but it's after the production of a zygote where the sperm and egg have fused together. So here are our prezygotic reproductive isolating mechanisms. This means we are isolating genes that will not be passed on through reproduction because of these different isolating factors. So our first one is geographical isolation. And that occurs just in cases when species exist in different areas, so they can't reach each other to breed. So they have been separated by geography. Also, there's ecological isolation, which results from two species who occur in the same area, but exist in different portions of the environment and are unlikely to hybridize. So even though they're on the same continent, they're separated by topography. They're not in the same habitat. So geographical means they can't reach each other at all. Ecological isolation means they can reach each other if they really try to, but it's highly unlikely because they just like different habitats. Also, there's temporal isolation, which results from two species having different reproductive periods or breeding seasons that preclude hybridization. So basically, they're not breeding at the same time, so they're not going to reproduce offspring together. There's also behavioral isolation, which is possibly the most interesting. It refers to the often elaborate courtship and mating rituals of some groups of animals, which tend to keep these species distinct in nature, even if they inhabit the same places. So this is like a bird that even though they could mate with any of the birds around them, they only want the bird with the specific song that they like. So that's behavioral isolation. They have expressed their preference for behavior, and that's who they will mate with. There's also mechanical isolation, and that means there is a physical barrier that is preventing reproduction. So it results from structural differences that prevent mating between related species of animals and plants. So the example that we had, well, we'll finish this slide. So then there's prevention of gamete fusion, which blocks the union of gametes, even following successful mating. So they could have had intercourse, they could have gotten together, but that zygote is not going to be formed because there is a blocking of the fusion of those gametes so that no zygote can be formed. Those are the prezygotic isolating mechanisms. And here are some of the examples that we went over. So we talked about how even though these same frogs are in the same pond and could possibly mate together, because of temporal isolation, they have different breeding periods. So they're not ready to breed at the same time as each other, so they won't. And that means we've got some reproductive isolation that's occurring because of that breeding timeline. It's temporal. Then there's ecological isolation, like the lion and tiger. They could reach each other if they wanted to, but they'd have to leave their habitats that they like. We also have behavioral isolation, which is like the cricket mating song. So we have crickets that only want crickets that have their specific song that they like. And then our bush babies have genitals that will only allow them to reproduce with each other if they are able to reach successfully. So because of the way they're designed, basically if it's with the wrong mate, they won't even be able, their genitals will not be able to even reach the other. So that's a mechanical isolation because it has a physical reason has stopped them from mating. They want to, they're both interested, but physically it's not going to happen. Aside from that, we have our post-zygotic barriers, which, and for our purposes, really, we're just talking about post-zygotic barriers, meaning a zygote was formed, but it will not be viable. So that's really all you need to remember. But a great example would just be mules, because they have this hybrid infertility, where even though the zygote was formed in their offspring, something happened to make that inviable or infertile, so that they will not be able to, their offspring will not be able to produce offspring. Some more examples that we have here. So let's see, is there anything new on this? Nope, we've got our bush babies, crickets, lions, frogs. Well, we could do some of these post-zygotic isolating mechanism examples. This is like the frogs of the genus Rana that can form hybrid tadpoles, but those tadpoles will die before adulthood. So they made a zygote, yes, but all of the tadpoles died before adulthood. Also, mules are the sterile hybrids of a male donkey and a female horse. So just kind of like the liger, the donkey and the horse were able to produce offspring, but that offspring is not able to produce offspring. And then hybrid breakdown. So the offspring of hybrid copepods have a reduced potential for survival or reproduction. So that just means that even though the zygote was formed, that offspring is not going to survive to the point where it can then reproduce. Now we're on to chapter 15. This was a short chapter on how we name living things scientifically. Anytime we're talking about the naming science, that's taxonomy. So taxonomy is the science that deals with the naming and grouping of organisms. And whenever we want to talk about studying organisms, it's necessary to give them names. This has been going on for many, many years. Biologists use a kind of multi-level grouping of individuals, and that's called classification. So we are classifying all these species into different groups. The earliest classification scheme grouped animals and plants into basic units called genera, which is just the plural for genus. And early biologists added a series of non-standardized descriptions to the genus name. This created a string of subjective names called a polynomial. So we used to scientifically have a lot of polynomial names, and they were getting kind of out of hand because they are so long. So here's an example of polynomial nomenclature. This was used in 1600s and on, and it would put similar species in common groups called genera, the genus, and each species was given a long name. So they had the polynomial, which was the genus name plus several descriptive words that fit whatever that species was. So for example, catnip, which is a plant, the polynomial name would have been Nepeta floribus interrupte spicatus pedunculatus. And imagine if we still named everything like that. Oh my gosh, how much worse would this class be? So the good news is they said, we've got to have a better system. We can't do this polynomial nomenclature. The names are getting out of hand. So enter Carolus Linnaeus, who came up with binomial nomenclature. And this is the standard that we are still using today for naming species. So he also grouped similar organisms into higher level categories based on similar characteristics. But he made it simpler. So if we've got a common name of tiger, instead of it being Panthera blah, 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 which would have been the polynomial nomenclature name, you can quote me on that. Instead, we have this nice, easy to follow two-part, which makes sense, binomial, two names. So all of our scientific species will have these two names. We've got a genus name, which is broad, and a species name, which is more specific. So there are different kinds of animals in the Panthera genus, but this is the tiger. So this is the Panthera tigris. Also, it's worth noting that whenever you're using binomial nomenclature, a scientific name is always going to be two-part. The first part is the genus, which is always capitalized. The second part is that more specific epithet, which is referring to the particular species, and it will not be capitalized. The two words together will be written in italics, but a lot of times if you're writing by hand, you will underline scientific names because it can be hard to distinguish what's italicized and what's not in handwriting. So yeah, our scientific name is binomial nomenclature. It's a two-part name per species. The first part of the name is capitalized, that's the genus. The second part of the name is more specific, it will be lowercase, and it is the species. If you're writing this by hand, you would probably underline it. If you were writing it on the computer, you could do italics or underline, either way. So our binomial has been expanded into a Linnaean system of classification. And this is just from the most general down to the most specific how we are classifying our different species on Earth. So first is our domain, next is kingdom, then we have phylum, the plural is phyla, then is the class, order, family, genus with a plural that is genera, and species. The plural is still just species. So we talked about our kingdoms of life in the very beginning of biology this semester. And multicellular, so we've got these 6 kingdoms. So we've got Animalia, which is the animals that we think of most often, fungi, eubacteria, archaebacteria, which you could think of this as bacteria and archaea as far as how we talked about it in the beginning of the semester, and then protista and plantae. So these are our 6 kingdoms that we're functioning with. And of these, all of our unicellular organisms will be in these 4 groups. So protists, fungi, eubacteria, and archaebacteria, all of these could have unicellular organisms. But everything that lives and is part of the plant or animal kingdom, Animalia or Plantae, those will all be multicellular. So we don't have any unicellular animals. We don't have any unicellular plants. There are unicellular fungi like yeast and unicellular examples of all of these. But plants and animals are all going to be multicellular. Whenever we're classifying living things, if you need to remember the order that we are classifying them in, from most general, meaning most inclusive. So the majority of things are in this, and then it gets more and more specific and more exclusive as we go down to species. But if you need to remember the order of that, they use this, King Philip can only find green socks. So that helps you to sort out from most general to most specific. If you can remember that King Philip can only find green socks, that gives you the order from kingdom to phylum, class to order, family to genus to species. So if we were looking at the hierarchical system used to classify a gray squirrel, just by this domain, kingdom, phylum, etc., this is what that would look like. So you can see we're very general when we're just in the domain of eukarya. And then we go on to the kingdoms. And this kingdom specifically is Animalia, because it's we're not focusing on a fungus or eubacteria, archaebacteria. We're focusing on an animal, so we're in the Animalia kingdom. After that, we go to the phylum of Chordata, which all of these animals exist within. This breakdown has subphylum, but we're not really focusing on that. So we'll go on to the next class, which is Mammalia. So these are our mammals. After that, we get to our order. So yes, it's a mammal, but what kind of mammal? This is our order Rodentia, which means it's one of the rodents. And then we get to the family, which is even more specific, and that's Scleridae. I can't say it. Scleridae. Sorry about that. And so that's just all of these different types of squirrels in this rodent order. Then we have our genus, which is Sclerus. So it could be either of these two types of squirrels, but once we get down to the species, that's very specifically what this organism is. And that is Sclerus carolinensis. So that just is an example of what you do with one species when you're trying to break it down and get more and more specific to name it and classify it. So looking at these classifications, which phylum do birds belong to? First, you would want to look for birds and then see what phylum does that fall under. Birds are in the vertebrates phylum. So these are all animals that have a backbone. And birds are one of them. And then it would get more specific as you go on to order. Which class is an eastern gray squirrel in? So we just talked about squirrels. We know that squirrels are mammals. So mammals would be the squirrel class. Also, arachnids and insects are two separate classes, but they're both part of which phylum? Arachnids, insects, which phylum do they come from? Arthropods. So they're from the Arthropoda phylum. Phylogeny is just the evolutionary history of an organism and its relationship to other species. So when we're talking about phylogeny, it's just what is the evolutionary history of this organism? And how does that relate to the other species that developed along the way? Scientists use different approaches to construct phylogenetic trees, which is just a way to trace that evolutionary history. Cladistics is an approach that infers phylogeny according to similarities derived from a common ancestor. So it infers, we're just like, we are assuming the phylogeny based on similarities that we're observing from a common ancestor. So derived characters are characters that are present in a group of organisms that arose from a common ancestor that lacked the character. A clade is a group of organisms which is related by descent that shared a derived character. So a clade is just a group like all of the organisms that received this specific type of fin or this specific kind of organ. Those are all in a clade together because they all received that characteristic from one ancestor, and that's how they got their derived character. By examining the distribution of derived traits among related organisms, it's possible to construct a cladogram, which is just a way, it's kind of like a family tree, but an evolutionary tree. It's a branching diagram that represents the phylogeny. So here's an example of a cladogram. And you can see by examining the distribution of derived traits among related organisms, it's possible to construct a cladogram, a branching diagram that represents the phylogeny. And this can all be guesswork just based on the kind of character that was observed. So if we went from bipedal organisms to organisms with no tail to organisms with hair, and that being the evolutionary branch where suddenly now organisms have hair, then we get to organisms that have an amniotic membrane. Now we're starting with organisms that actually have lungs, back to the evolution where jaws were developed after lampreys. So this is just a way to trace evolutionary change in kind of a family tree. But what's the difference? Because we've talked about cladograms and I mentioned phylogenetic trees. And it sounds like both of them are tracing these evolutionary relationships. So what sets them apart from each other? Well, a cladogram, the relationships are all hypothetical. You could sit down and make one right now based on when you think different evolutionary changes happened, based on the first time an animal with hair came about or the first time fins were documented. You can make your own. It's guesswork. But phylogenetic trees are like the science-backed version of that. So these relationships are backed by molecular evidence that has been studied, and they should have access to DNA other than molecular data. To DNA or other molecular data. So that's how they're tracing them.